All right. Welcome, everyone. I'm super stoked for the seminar series and even more stoked because it's my pleasure to introduce Dr. Archana Anand today, who's going to be our first speaker for the CSU webinar series. Fun fact, we went to high school together back in India, and then we both had very different career trajectories because we both got degrees in engineering and computer science. And then, Archana got an MBA as well, right? I think, right? But then she's going to tell you all of that. I don't want to preempt any of her fun stuff. But Dr. Anand is an assistant professor in the Department of Biology at San Francisco State University. She received her engineering degree from the National University of Singapore and then a PhD from the University of Hong Kong. And then she spent 1 year as a postdoc or 2 years as a postdoc at Columbia, followed by another postdoc at Stanford before she started her faculty position at SF State. I had the pleasure of visiting their lab and at SF State earlier this year, and it was amazing. And it's always so cool to learn about what everybody at the CSU is doing. And I'm really hoping that this is our opportunity as a group to focus and highlight all of the cool things that we as CSU folks are doing in the bioinformatics field. So with that, without much further ado, take it away, Archana.
Thank you very much, Arun. Nice to see you again, Scott, as well. I was part of the CSU Bioinformatics series last semester, I believe, and it was wonderful. So it's a wonderful initiative that you've started, and I'm glad to be a part of it. So today I want to take you through a few stories, and they will all be, the idea is, of course, for me to unveil some stories through bioinformatics, and whether that'll give you some insights on diverse microbial communities and different ecosystems. So without, let me get into all of that, because I think a little of my background will come as I reveal those stories to you. So I want to first begin by saying that bioinformatics has power. It's a statement that I'm sure you know already, but I want to highlight how it serves as this bridge between data, actionable insights across different biological systems, challenges, marine ecosystems, and even human health. So it's a fascinating toolkit that we now have at our disposal. And I want to take this opportunity today to take you through three stories. And as a scientist, we're all encouraged to ask research questions. So what's an experiment without a question, right? So the stories that I have are these questions. So the first thing I would like to talk about is how microbial communities contribute to the health of ecosystems, such as oyster reefs. And then what happens to marine ecosystems when they are exposed to contaminants, say from wastewater? And I'll end by talking about how can we detect the presence of antimicrobial resistance in our environment before it becomes a public health crisis. So I'll start off with story one. And before I begin, I want to acknowledge my funding sources, the Department of Biology at San Francisco State University, NSF, Center for Cellular Construction, NIH, and there's a Chan Zuckerberg BioHub contribution to this effort as well, which we're very grateful for. And I have some photos of students who have participated. There are several others that I just haven't received headshots from. But I am so grateful for the wonderful team that I have here at SF State. So let me begin. This is, oyster reefs, as you could tell from the question, they are invaluable ecosystems, and they provide a wide variety of critical ecosystem services. They filter water, they protect coastlines, they provide habitat protection from various marine species. But these habitats have been facing escalating threats from climate change and human stresses. Recently in San Francisco, the Bay Area, we experienced a huge atmospheric river event. This was in this current wet season that we've just gone past. And this, we don't know, it could have driven several oyster die-off events that we have seen in restoration sites across the bay. Now, for those of you who are not entirely familiar, oyster reefs are primarily formed by shells. They are formed by the accumulation of these oyster shells, and these are aspects that are not yet fully understood, but when they do have a swimming phase in their larval stage, after which they would need to find the perfect substrate and settle on them, after which they stay stagnant, so they do not, they are sessile through the rest of their life cycle. So the motile and the sessile stages encompass the entire life cycle of an oyster. And to date, the cues that the adults, the larvae feed into in order to make some decisions to settle on a certain substrate are unknown. So right now there are several initiatives that look at physical and chemical choices for substrates. And some of them even throw oyster shells that are of dead adult oysters. So you may have already heard of this. They put in shell bags because the biological cues that are brought about from the adult oyster shells could still facilitate in larval settlement in the substrates is the idea. But to date, we still don't understand whether microbial communities play a role and what microbial communities are present in these invaluable ecosystems. And we know this to a certain extent based on some research that's been undertaken in other parts of the world. So I know University of Melbourne, Rutgers University is doing some fascinating work. In Asia, University of Hong Kong has also been doing a lot of work on oyster restoration. In the western coast of the United States, we do have some work, but there is still the understanding that we have on how the contribution of microbial communities to the restoration success of oyster reefs is very fragmented. So the native oyster that we have here is called, oh, sorry, are you able to see? Yeah, are you able to see my screen on the map? Because I think I lost it for a minute, but should be back now.
Yes.
So the native oyster species that we have here is called Ostrea lurida, and it's a small oyster species compared to the other Pacific oyster, which is relatively large in body size. And the study that I'm going to share is being led by a graduate student, Jeff, in my lab, and in collaboration with several other students whose photos you've also seen. And they've all worked fantastically in developing a protocol to collect different samples and figure out how we could even do something like this because it's the first of its kind here. So what you see on the right side of this figure is 4 sites. You see Brickyard Park, Dunphy Park, and Heron's Head at the bottom. And at the very top, you see Point Pinole. Before I begin, I want you to know that in the atmospheric river event that I spoke about recently, Point Pinole saw a big die-off, and we witnessed extensive oyster mortality there compared to the other 3 sites. We do not know if that was the direct cause, but it just happened that we were sampling around that period of time. And so this may have skewed some of the results that we see as well. So before I get into the bioinformatics itself, I think I should just give you a little more context. So do bear with me. What we did was we looked at 3 different things. We looked at oyster, sediment, and biofilm. So oyster, when I say oyster, we looked at the tissue inside the oyster. Of course, we were limited in how many oyster individuals we could collect because we were conscious about these die-off events that had affected these restoration sites. So there were times when we did not collect any oysters at all. We didn't find any that were viable. Sometimes we collected just one individual and we made copies of them for DNA extraction. Sediment and biofilm we did do. We scraped surfaces of oyster shells and we also scraped surfaces of the pilings that were artificial substrates that have been put there by the Smithsonian Environmental Research Center and the National Estuarine Research Reserve. So these were the 2 institutions that collaborated with us, and we are very grateful to them for having us be part of their sampling efforts. And our question was, what microbes live there? So to do this, I want to introduce one bioinformatic tool as part of this talk, and that is 16S amplicon sequencing. Now, 16S amplicon sequencing, for those of you who are not familiar, this table should, if you rest your eyes on it and ignore what I'm saying, that's completely fine. And it is the goal is for us to look at what bacteria and archaeal species are present in a community. It's very specific to those two kinds of groups. And what we do to find them is we use the 16S ribosomal RNA gene, which tends to be very conserved in both bacteria and archaea. And although it does have some conserved and variable regions, we pick a certain region that we think would work really well, depending on how well curated the databases are. And this allows for the identification of microorganisms across various taxonomic levels, but within bacteria and archaea. So that was what we were hoping to do. Of course, you can argue that we need to look at other things if you are already heavily into bioinformatics, but we thought this would be a good start for us to build a baseline of the environmental microbiome within oyster restoration sites or oyster beds in the West Coast, in this part of the bay. So I have two slides that is just going to show some data to you so that you could marvel at the power of bioinformatics again. So the first of this is on the relative abundance at the phyla level based on sample type and location. So this is a busy figure. So let me help you look at this. So we had three different types of samples, as I mentioned. First is biofilm, oyster, and sediment. And within the sample types, you see the different sites. So if you recall, I mentioned 4 sites that we got the samples from. Only for sediment did we get the data from all the 4 sites. So these sites are listed not in the order I think that you see, but Point Pinole is up at the very north, Heron's Head is in the south, and Brickyard and Dunphy are in the middle. I guess we could have arranged them for you in the order of the map, so it might have been easier. For biofilm and oysters, we didn't get some samples, and we didn't have the 16S amplicon sequencing data failed. It could have been an issue with our pipeline as well. So this is something that we have to look into. So I do not present data from the sample that didn't work. And that's where you see just 3 sampling locations. What we did see was that the biofilm samples were distinct compared to the oyster and the sediment. So that was the biggest takeaway for us from this. And it was a bit interesting because the oysters would tend to exchange water and they filter water, right? So they're usually found mucky in and around sediment. But it was interesting that the biofilm seems to be the one that was visibly distinct compared to the other 2 microorganisms that were present within the oyster as well as outside the oyster in the sediment. And this figure shows you a differential abundance analysis. So the goal of this talk was, of course, not to confuse you, but the main goal is to show you that once you have bioinformatics data, that you could use the data and do additional analysis on that to help reveal things that might be, that would make a fascinating story. So in this case, here, what you see is the differential abundance of some taxa in biofilm compared to oyster tissue is what you see here, which is why everything in biofilm is highlighted in peach versus there are some that are highlighted in purple in the oyster. So I don't want you to look at the sites that on the left, the 2 columns that have site at the top, but just to focus on the sample type. And what we did see was that, like I said, biofilms were distinct, but we also saw that the biofilms didn't have microbes that were all necessarily considered to be good. So there were pathogenic microbes present, like streptococcus. There were some, and these could be because of human presence in the site. It could be because of runoff, that the sites are non-point runoffs that we're not entirely sure of. From sewage treatment facilities or from farms. So these are things that we still don't yet understand fully as to why some of these pathogens might be present. We also, like Neisseria is also another human pathogen. we also found some probiotic bacteria. And these probiotic bacteria, recently there have been studies that are showing that oysters make probiotic, or they also facilitate the growth of some of these probiotic organisms. They make probiotic enzymes that facilitate their attachment to some substrates that they like versus others, and therefore promotes resilience to disease or colonization resistance. And again, these are just preliminary data. This is from a preprint. So I will stop right there and move on to my next story before I get into it too heavily. And if you have any questions, feel free to put it in the chat and I will get to them towards the end of that talk. Okay, so the next thing I'd like to talk to you about is I'm going to switch gears a little bit, and we're still staying with marine ecosystems, but I'd like to talk about what happens to them when they're exposed to contaminants from wastewater. And to those of you who already know me, you know I love wastewater, so it has to be a part of any talk that I'm giving, and which is why it's making its appearance here. A lot of this work is results from at least the first part of it will be from my PhD and in Hong Kong. So I'd like to acknowledge the funding agencies and all my collaborators at Hong Kong and my lab mates who I'm still in touch with. So I'll give you some context as to why I'm talking about wastewater all of a sudden, because we were just talking about oyster beds in San Francisco, and now we're in Hong Kong. So this slide should help you understand that just 10 rivers carry over 90% of all the global marine litter into the oceans, and 8 of these rivers are in Asia. But something that's even more shocking is that even today, over 90% of all the world's wastewater that gets dumped into the ocean is untreated. And most people don't know where their sewage goes. They don't wonder what happens after they flush the toilet and where the sewage goes in those pipes. So when I learned about this, I was interested in looking at how this could affect marine life. And you can ask me why I was even interested in something like this. And this is because you see in this picture, the photo on the right shows you Hong Kong. It's a highly urbanized city. It has a population density of over 7,000 people per square kilometer of land. Compare this with, say, Iceland, that has a population density of about 3 people per square kilometer of land. But despite the presence and the pressure from urbanization on the surrounding environment, Hong Kong is a marine life powerhouse. And I say this because it has over 80 species of hard coral, which is even more than what you can find in the Caribbean. Did you know that? So the next time you think of going to the Caribbean, you could visit Hong Kong instead and see way more hard corals. Well, so I was interested in looking at what happens to the marine ecosystems. And there were plenty of teams that were looking at charismatic organisms like the Chinese white dolphin, the finless porpoise, but there was not much attention being given to marine microbes at that time. So, and yet they occupy about 70% of all life in the ocean. And even today, I think over 70% is either unknown, unnamed, or invisible to the naked eye. So that should hopefully give you a rationale. And I began to lead a project called the Marine Global Earth Observatories Program, Marine Geo. And this was in collaboration with the Smithsonian Institution. And they were hoping to set up a site, their first site in Asia. So Hong Kong was the first site. And what I'm showing you now is a map of Hong Kong. So this is Hong Kong's special administrative region. And I have the bar of zoom right on top of the map. I'm sorry, I'm just moving that over. So this is Hong Kong. And what you see here is a zoomed version of that. And the sites that you see here, which are labeled in red, correspond to these sites at the bottom, which have where they're classified as high environment or high pollution. Then there's one site which is considered to be a medium site or medium impacted site. And there are 3 teal sites. And those are considered to be, again, low impact sites. And why we classify them is something that, yes, you could, again, challenge. And these were we have environmental data that goes back to the 1980s. The Hong Kong government is amazing. They've collected this data through 70 monitoring stations across the marine environment. So these were all these variables that we got the data from, and we used this to classify them in this manner. And like I said, of course, you can challenge them, but we thought this would be the best fit. And we use these mini ocean hotels. I love to tell the story, so some of you might have heard this, and if you did, please bear with me. I have different results to share. So these are PVC plates that are stacked one on top of the other. There are 9 PVC plates per ocean hotel. Their actual name is Autonomous Reef Monitoring Structures, ARMS. And I like the term mini ocean hotels. We leave them in the ocean floor for, I hope you can see this. We leave them in the ocean floor for a year or two, after which we collect and colonize everything that's, we collect and identify everything that's colonized these plates. So what you see here is me with my dive buddy, Mike. What we're doing is we have placed a white bucket on an ARMS unit that is ready to be retrieved. We've secured them using bungee cords. And the reason we're doing this is that we are interested in the microbial communities that are not just sessile, that not just the ones that choose to settle on the plates, but they could be present immediately surrounding those plates, but maybe motile. And their signatures could be derived by filtering the water that's just around the arms. So which is why we collect the water as well. I don't know how I guess I can't fast forward it for you, but then essentially we retrieve, we bring it up, we take it to a boat, and then we disassemble the arms unit, and we look at everything in the plates. So we take pictures. I think something is stuck, which is why you might think we're not moving much. And we're probably trying to retrieve it from the bottom. So once we take it out, when we take pictures, we sometimes take these organisms, which are so beautiful. So I just wanted to show you, give you an example of what we are likely to see. And we can use different tools to look at the organisms there. So now I'm going to come back to the bioinformatics aim of this talk, and introduce another tool, which I'm not sure if you're familiar with, but it's an age-old tool. It's called meta barcoding. And in this case, we choose biomarkers or barcodes, and CO1 is the barcode that we chose. Now I'm going to take a moment to kind of explain what this is to you, so you feel a little better about the whole thing. So this is a molecular technique that's used to identify animal species in a mixed environmental sample. And I say that because what we do to the arms plates when it comes out is it's really hard. It would be the result of several organisms settling on them over, say, 1 to 2 years. So most of the time we end up using these hardcore scrapers to scrape them off, which means I'm sure all the DNA would get mingled up. And then sometimes we put them through a blender to homogenize the sample, so that when we pipette out a certain quantity for DNA extraction, we get a homogeneous or a composite sample that is likely to be representative of everything that's in there, or as much as possible. So that's why this marker is meant to give us a sense of everything that's in a mixed sample. And what we do is these are also conserved within broad taxonomic groups. So unlike 16S, which only focuses on the bacteria and archaea, CO1 tends to give you eukaryotes. So you're likely to see many more animals of a species in there. And we usually tend to amplify them using PCR, and then followed by Illumina sequencing. Now, this is something that we did because at that time, meta barcoding was relatively less expensive when compared to 16S or shotgun sequencing. So when you take on these projects, again, budget is, of course, very important. So that would play a role in your decision as well. And this was our effort in building the first genetic library of marine life in Hong Kong. So, which is why we started out with the most basic fundamental thing that we could think of and then went from there. So, having given you the context of how Hong Kong is in terms of its population density, you might think of some things, I'm sure, or maybe. And what we found was really interesting because we and this was completely unexpected because we had taxonomists coming in from the United States. There were some from the Middle East, from the Red Sea, and they've all seen they are used to seeing tons and tons of marine species richness in very pristine sites. And when they flew in, they said, "Oh, this is such a polluted site. We're unlikely to see much." And they were shocked that the South China Sea, which is what we represent from our sample, was skyrocketed, the OTU richness table, when compared to the same, compared to results obtained from the same sampling methodology in the Red Sea, in Florida and Virginia, and even in Singapore. So this is something that we just published this paper. We had to go back and forth and look at the data again and again because we weren't sure if this was, really the case, and we were all shocked to see this. And it's something I'm going to speak about this a little bit more. But then what was happening was there was something called environmental filtering that was happening because of urbanization pressure. And it tended to skew the samples towards one site over the other. And that happened to be the site that had the medium impact and not the high or the low impact site. So again, this was interesting because you will expect the site that has low environmental pollution to also have the higher biodiversity. But that wasn't the case. And this followed something called an intermediate disturbance hypothesis, which says that a site that is not a site of high disturbance and not a site, neither is it a site of low environmental disturbance, but a site of medium environmental disturbance is the one that is likely to provide the best environment for higher biodiversity. Yes, so that was a fascinating study. Now, I also want to take you through another study that will be used to introduce you to another bioinformatic tool. And this is also in Hong Kong. The study just came out as a paper much earlier, but we did everything at the same time. So this should give you a sense of how publications work in academia. We focused on 4 sites. So this is me with my dive buddy Taehoon. What we're doing is burying tea bags, and this is not what I'm going to be talking about today. I'd love to talk about it outside of this meeting, but what we are doing is we also collected sediment from the same sites that we buried the ocean arms. The study that I'm going to share with you is from these 4 sites that were in a harbor called Tolo Harbor. And this was a harbor that is notorious for wastewater pollution, because if you see here what is denoted as number 3 and 3 on the 2 ends of the innermost part of Tolo Harbor were 2 gigantic wastewater treatment facilities that would dump untreated wastewater, and then it was soon upgraded in the 1990s into Tolo Harbor. So this is almost like a natural laboratory as you move from Center Island to Chele Pai, Port Island, and this is Tung Ping Chau that you cannot see here. As you move from Center Island to Tung Ping Chau, there is significant improvement in what you can visibly see as marine life. So you'll get to see almost no corals at all in Center Island. You might see about 10 species, you see about 30, and then at Tung Ping Chau, you see about 60 species of hard coral. So that should give you a sense of other environmental parameters there. And so we wanted to look at marine sediment, and this is because sediment has tons of microbes. It's one of the most diverse in microbial assemblages and function. They're also very understudied. It's also difficult for us to extrapolate what we find in one place to other places. They sequester carbon. So there were several reasons for us to say, okay, let's look at the sediment in these sites. So now what I want to introduce you to is another tool called shotgun sequencing. And you might have heard of this term, or it might be called metagenomics or whole genome sequencing. They're all the same. And the goal of this is to extract the total DNA in a sample. So which means it'll include the DNA of all the organisms. It could be bacteria, archaea, viruses, fungi, plants, animals, everything in there as much as we can. And then, which means that when we get these genomes, we're likely to also get function. We'll also get genes that will help us figure out what they are doing in that place. So at this point, I want to just stop for a second and just go back to what I just shared, which was I first spoke to you about CO1 and 16S. And what those two will tell you is who lives in a certain place. And that's what they'll tell you. They'll give you their genes. You could infer that they are doing something based on the genes that they provide, but it stops right there. What shotgun sequencing can tell you is it'll tell you who lives there and a little more about what they're doing there. Again, it's still an inference because this is not RNA sequencing. So we're not going to be, I cannot claim that they are doing this. But we'll get enough information about the genes for us to figure out something like this. So in the scientific community, heat maps are very popular. It's also very useful. It's a wonderful way to represent some things pretty in a simple format as well. So what this shows you is on the left, the CI, CLP, PI, and TPC are Center Island, Chile-Pai, Port Island, and Tongpingchao. So they're from the most polluted sites here to the least polluted sites on top. And we saw that there were genes that were activated for different functions, but it wasn't a particularly stellar, we couldn't tell like a lot. We could say that there were some genes in Center Island that were more pronounced than, which at least were, the gene count was higher than what it was in Tungping but we did see that overall, there were some functions that could have been associated to metabolic pathways in urban environments, in polluted environments that we were seeing in Hong Kong. So that was as one of the messages. And then what you can also see is things like a shift in the metabolism between sites. So this is, again, something that you can do using bioinformatics, but you would also need an additional step, like I said. So you use the bioinformatics data and then maybe work on the data using like a differential abundance analysis, which I like. And it'll give you, in this case, it gives you the top 25 abundant families here. These are the taxa that are at the bottom, the legend. And it'll show you what those top 25 most abundant families contribute to in their metabolism when you compare Center Island versus Tungping Chao. So Center Island is the case group and Tungping Chao was our control because Tungping Chao, we're considering that to be relatively more pristine. But of course it's not, because it's all in a highly urban impacted environment. So in the interest of time, I'm just going to move on to my last story. So please park your questions in the chat or write them down, and I'd love to talk more about it just at the end. So now I'm going to, again, switch gears, but hopefully it's all still relevant and connected because there's a wastewater theme tied to it. How can we detect the presence of antimicrobial resistance in our environment before it becomes a public health crisis? Now, this has been, again, I'd like to, of course, acknowledge all my students. And there are several of them who participated. And I'm just going to mention their names as we go from left to right. So there's Ben, Gabby, Jeff, Michael, Kate, and Michael. And there are several others whose photos you don't see. And so I'm really, really grateful for a wonderful lab that I'm blessed with here at SF State. But I also want to acknowledge the supporters, our collaborators, and funding agencies. So I'm just going to spend one slide on how this all began. So I think Arun mentioned that I was working on, I did a postdoc at Columbia and Stanford. So I was part of one of the first few efforts that looked at SARS-CoV-2 in wastewater, and we were trying to figure out, is SARS-CoV-2 in wastewater? Can it be even correlated to these clinical cases? Can we use it to tell counties and ICUs that, prepare the ICUs for more beds because we're seeing like a spike in infection rates based on what we see in the wastewater? So yes, we did that. And what we also saw was we could see mutations in the amino acids within the SARS-CoV-2 genome. So now all the other tools that I mentioned, it's similar to that. We did something, this application is just variant calling in bioinformatics, and I'm not going into it, but I'm mentioning it because I'd like for those of you who are attending who are interested in it to reach out to me, and I'll be happy to direct you to resources on that. So it was just to look at mutations within a genome and see if that could help us understand whether a virus is more transmissive or more infectious or more virulent in a population. Now, at SF State, I am now part of a phage foundry effort. This is funded by the Department of Energy. This is in collaboration with Berkeley, University of Texas, Chicago, and the Berkeley Lab, which is a national lab. And what we have done, and this has been led by my graduate student, Michael, what Michael has done is he has developed a protocol to extract DNA and RNA from wastewater, but using magnetic beads, which is proven to be more efficient and sensitive, and also use that to help us understand not just microbes. So there's just looking at viruses, there's looking at other microbes, but to understand how microbes interact and whether that can tell us something about whether there are viruses that are regulating the populations of bacteria there, or fungi that are working with the bacteria and therefore regulating some disease outbreaks. So there's much more that we can look at right now than just SARS-CoV-2. So what bioinformatics tool do you think I will likely need to look at antimicrobial resistance genes in wastewater? I've introduced just 3 tools, so forget what I said about variant calling. And for some reason, I can't see my chat box, but, oh, I can see it now. But if yes, I'm just waiting to see if there are others who have thoughts on this before I go on. I only have a few slides, so we can spend a minute having, you can paste your comments in the chat. Well, I see Arun and Scott's remarks, and yes, so yes, and a really good database of ARGs too. That's very important. So yes, I've listed a shotgun, whole genome, or metagenomics sequencing. These are well, they're all one and the same. This is what you'll need, and I want to, at this point, introduce 2 databases to this group because I didn't do that the last time I was here. And I thought if this is a growing audience that is really interested in bioinformatics, which I can tell you are, I want to introduce you to 2 of these databases. They are free to use. They are user-friendly platforms. They are web-based, which means you do not need to code when you throw in your FASTQ files into these platforms. The CZ ID, you can look it up. It's a Chan Zuckerberg ID platform. It is meant to give you shotgun. They'll do a shotgun sequencing on your FASTQ files. They will do the assembly for you. And I'm not getting into the nitty-gritties of what the pipeline is, but it would give you something that looks like this. It would give you a heat map, and you could click a filter and say, look, I want to look at just known pathogens. And it would give you a list of what it has been fed by, of course, the data scientists who are building the platform as known pathogens at that time, and provide a heat map for you. So what you can see here in this figure is influent, so which means it's wastewater that has not been treated. So just after it leaves the toilets in the pipes, reaches the wastewater treatment facility, but has not been treated just yet. This is from the wet season on the left, and this is from the dry season on the right. And every column of cells is one sample. So which means in the wet season, we had so many samples, and in the dry season, we had so many samples. So you can clearly tell from this figure that there are already some pathogens that are more prevalent in the wet season than it is in the dry season, right? So I'm hoping you're able to see it's more red in the wet season than it is in the dry season, as you would expect, because we also get the flu season is coming up, and we tend to all fall more sick in the wet season than we do in the dry season. So this is to show you the differential abundance of wet versus dry season and again, AMR genes in these two seasons. And the Chan-Zuckerberg ID pipeline also spits out AMR genes to you. So it would give you a CSV file that you could then use to make some simple Excel plots. I see Scott's put in another database as well that could be useful. There is an NCBI AMR finder. So yes, and this, if there's any, again, questions about this, we can always have another discussion or an exchange after this talk as well. So there are, as you can see, there are some of these genes, and I have a slide that will help you understand what these are, because these are not English, they're just alphabets to us right now in this slide. And carbapenem resistance genes by species, this is something that has come up extensively now because of antimicrobial resistance. There's also, there was an article that WHO released, and they mentioned that this resistance is a threat for public health, and especially because it's pronounced in pathogens like Klebsiella, which cause a respiratory disease, severe respiratory disease. So this shows you the resistance that we found across some species. And what it also helps you, what it gives you is whether the gene is in the chromosome or in the plasmid. So you can tell if it's integrated in the genome or whether it's separate, which will tell you something about the host and the pathogen. This is my last slide before I get to conclusions. So I just wanted to show, elaborate on some of the abbreviations that you saw on the previous slides and what those genes could mean. Because some of us would be familiar with these antibiotics like erythromycin, for instance. And if you're interested in knowing what antimicrobial resistance gene marker you need to look for, it's the MSRE marker that I'm highlighting here. There are several other resistance gene markers that are associated with, say, UTIs, which is a big deal right now. There's a lot of research going into that because of problems with resistance. So I'm not going to go into too much of that. I want to conclude by saying that bioinformatics is evolving, and I only spoke about the shotgun, metagenomics, and short reads. I did not get into long reads or PacBio and Nanopore, and that's a whole world out there. So as bioinformatics continues to evolve, something that I want to leave you with is, how do you think we can address continued complex challenges that are at the intersection of environmental and public health? So with that, I'm grateful for this opportunity to be here, and thank you for your time.
Thank you. That was fascinating. That was great. Let me look at the view here. Cool. Yeah, that was really fun. Such great stories, and like all over the world too, which is really fantastic. I have a bunch of questions that I've been collecting from my students, and I'd like to ask you some of them. They're really good. So let's, you don't mind starting. I'm going to combine a couple.
Oh, sure.
Similar. But so one of them from, I guess is from Lindsay and Wyman, a sort of combo question. How do you determine the genomes or the species in the shotgun sequences? Are they aligned to genomes or databases? And the follow-up question is, what happens if you can't determine the genome? Like you get in there, do you have things where you can't figure it out?
Yes, so to answer your first question, yes, there are databases. An example is the NCBI database for 16S. There's also the SILVA, SILVA database, which is now considered to be quite nicely curated for 16S genes. There are several others, and that would change. So the databases would change depending on the marker that you use to mine your genome. So they are very, very biomarker specific, if I can say so. And if you're doing amplicon sequencing, so there would be an ITS database, 16S database, 18S database, and so on and so forth. For the metagenomes, they do a blast as well on NCBI. It tends to be that way. And you could compare the 16S and the shotgun taxonomy and see, but shotgun data are usually considered very reliable when compared to the 16S because they give you much more. Unless you want just the bacteria and archaea, then yes, I would say choose the 16S. Maybe now I'm babbling too much. Okay, the next question is, what if it fails? I think we've had some problems with our soil data. We collected some soil in a vineyard, and I think this has been led by my student Gabby, who's doing a master's now. She's so I think she collects tilled soil, untilled soil, and she's been. We saw really short fragments of DNA, and that what happens is there's a filtering step in bioinformatics, and it doesn't pass the filter because the fragments are too short. So in which case, it doesn't mean that it has failed. You would just pass it, maybe skip a step, or you would declare that the fragments were of a certain length, which are shorter than what ideally the case in, say, peer-reviewed articles, and proceed with the workflow and still go ahead with it. So I wouldn't say there's any such thing as a completely failed. Yeah, if I can say
I
that.
don't know about that. I know that a lot of the databases, like, so if you have a random piece of DNA from a shotgun and you do not have a match in a database, you do not know what it is. And in the beginning, that was like 70%, and now it's gone down to 50%. And people just throw away the reads and they ignore them. So I don't know if we've gotten to a point where we can get about 70 or 80% of the sequences identified now, which I think is a huge, like in my lab, we're able to do that with assemblies and stuff. But yeah, sometimes you honestly don't have any idea.
Yeah, so I guess if it comes to the unknown, then I completely agree with Scott. We don't know, and it would just, that's because we have databases that just don't cover it, and we just don't know what they are. So we would classify them as unknown, as you would have seen in those figures. There are several unknown bars that would be much bigger than the other bars. And even for the AMR genes, we set cutoffs. So the cutoff is 80%. And 80%, I mean, it's not 100%. So there's still like, we do a lot, and then sometimes we don't find what we want. So I agree completely with Scott that yes, in that way, I guess you don't find everything.
Yeah, absolutely. Should I go some more questions here from the students? There's some cool, it's like, are wastewater ejection sites affecting the microbiomes developed on the coastal oyster communities? Do you know that?
That's a great question. And I would think that they are affecting coastal coastal marine communities for sure. We don't know about I can't say a cause and effect right now in this call because we just don't know. But as a general guideline, yes, we are seeing huge discharge outfall effects on marine life in the bay, even in South Bay. So that's one of the things. There's actually a hypoxic zone in South Bay. We're doing some eDNA work. Again, another grad student is doing that, Luz Maria, and she's been finding changes in the fish communities in the bay compared to not just she's also looking at the microbes, but then over time series, she's seeing some changes. So that's I hope that answers your question, although it's not definitive.
Great. This is an interesting one. How long does it take for the ocean hotels to be populated for, like, when they're complete so you're ready to sample them?
That's a really good
that process like?
That's a really good question. I think when we first started it, we just had like one guideline. We would do it for 2 years. And then there was we started another thing where we began sampling it for 6 months to look at temporal scale changes in the ocean hotels, exactly what you're asking, because nobody knew what the perfect time was. And we found that these hotels in tropical sites would get populated faster than they would in temperate sites. And again, there are some, I don't think much has come out from this because it's not just, we've not yet collated all the data because the Smithsonian, we feed all our information to all of them. And we speak to some of our collaborators. So I just know this from universities I work with. And yes, there are changes temporarily. There's no set guideline, which is why I said 1 to 2 years. So even right now, we do it 1 to 2 years.
Great. Thank you. And that was from Allison, and the previous one from Giovanni, as far as the questions go. This is an interesting one. What are the ethical, what are the ethical and ecological considerations with using this metagenomics or this DNA technology? Should you be, is it something to be concerned about? Like, is it really destructive, or is it, are there ways to, is it just, there's so much in there that this is such a minor impact that it doesn't matter?
No, I think there are ethical implications for all this, and I think it comes from the sample source. So I see it as two things. I think one is the sampling itself. So if it's wastewater, then in wastewater is good because it is anonymous and it's unbiased. So, and usually the population size is over 500, so you can't trace it back to anyone. So there are these ethical considerations that we have to make sure we pass. So you can't go to a house and sample wastewater because that would not be ethical without consent. The other thing, so usually when we do these wastewater studies, we go to an IRB, an institutional review board, and so far IRB in universities do not require the wastewater data itself to go through the process. But if we use clinical data to correlate the wastewater data with, then yes, it needs to go through an IRB approval, unless the clinical data is from a public dashboard like the San Francisco City of Public Health or CDC, perhaps. So that's, I hope that should give you some sense. Another thing that I think would be important to think about ethically would be what do you do from there? Like, because now what's next? And I think everyone's talking about engineering genomes, gene editing. And that, I think, yes, has significant ethical considerations that have to be kept in mind, because there are discussions about could we put in a substrate in, say, an oyster bed, and that would be fed with an organism that is proven to benefit the oyster reef. This would have several ramifications to other organisms in that ecosystem that we have not yet studied. So this might need an environmental impact assessment. I mean, yes, so I'm sure there are several ethical considerations that have to be taken into account.
That was great. I forgot to say that was Donna's question. Great. I had there's a bunch of other questions. We go on for a while, but I want to let people here ask questions personally if they have one.
I got one. I put mine in and this might be a question for both of you actually, because you two are sort of the experts in these kinds of analyses. So, are there sort of best practices with respect to dealing with pipelines with amplicon sequencing kind of data sets? And this comes from sort of like personal experience, because I'm dealing with some soil data sets right now, which, you run the same data against the same database using Chime, Mother, Data2, they all give you different results. And I'm starting to wonder what do I believe and what to not believe. So any thoughts on sort of best practices? Because I think that would be helpful for a lot of us who might be doing this kind of work. And I also want to be in a position to teach what would be the more reliable way. So any thoughts would be appreciated.
Chime2, definitely.
Yeah.
Everything through Chime2. Nothing. I'm an author on Chime2. So.
Right.
No, but it's interesting. Can I clarify? Like, so you say you run the same data. I totally get that. But all your like you could be running. You're running the exact same version of all the programs in every database, or this exact same. The database itself is going to give you something different. If you don't have the same bugs in two databases, you're going to get different. So I have compared directly Silva and green genes. And what I would do sometimes is take the top 3 hits of all of them, and I would say how different they are. And sometimes you get a difference between the top second or 2, and it looks a lot different, but it wasn't as different as I expected, even though green genes is really old. So I wonder if it's, you know, like ASVs is another thing. If you're using to blur, you're going to get a different result than data 2, which is another user. You're going to get a different result than there's another one, use search and everything, because it's the way that the filtering algorithm. So if you have a different ASV, you're going to get a different thing, and you're going to get a different counts. So I would say that there are I would expect, as long as you clean the data and it's exactly the same going in, you're talking about the denoisers being different, and you're going to talk about the database being different. And then you have to ask yourself, like, well, at what level does the difference matter? So you have to say, okay, well, I can't really get the strain information, but I could get the genus information maybe. Is that pretty similar? How's the families looking, you know? And then you have to say, yeah, you know, because the bigger problem you often have is if you tell 2 samples and you use a different DNA extraction method, don't ever do that. Because it's going to be like, it's just so biased. And then, or a different PCR primer set, you can't even compare them. So yes, these are all considerations, and it's like a rule of thumb. And you know what people don't do, and Archana will tell you this, is they don't go back and verify the results with a totally independent method like real-time PCR. So if you really give a crap about bug X, you need to go back and take some primers and amplify it directly out of the sample. And nobody does that because it's a pain in the rear. So it just depends on how fuzzy you want to get things, how close you want your questions to be answered. Archana, I'm sorry, I went on a rant.
That was perfect.
But yeah, don't trust it. Like, I think this is right. Like, our bioinformatics, don't trust it. Like, don't trust it. Verify it 3 ways to Sunday, do the stats test right, and then say, these are hypotheses that we're testing, right? I mean, there's mathematical things to think about. However, if we didn't do this, we wouldn't know anything. Nothing, because it would take you 1,000,000 years to culture everything out of these samples. Like, you'd never get anywhere. So, and I think the same with metagenomics. We do these assemblies, and sometimes they actually be assemblies of multiple things. Like, they might not be perfect. So do you go back and you verify that that's the strain you think it is? Do people do that? No, they don't do that. I've done it in some ways, and we could talk about Archana with bioinformatics. But I would go back and PCR those guys. Is that really the strain you think it is? Because I've had cases where I thought we had salmonella, and I'm like, well, maybe we do, but it's not that salmonella.
Yeah.
You know? Anyway, I'll let you ask. You continue.
No, I think that was good. Yeah, please go ahead.
No, I was going to say, you know, like you were just mentioning, right? I mean, because, you know, Archana, you had Neisseria in one of the biofilms
Yeah.
in oysters, right? And I, you know, the CDC reported like a whole bunch of like Neisseria outbreaks, but perhaps it's not the same species, right? So the one that was specifically that the CDC put out was a meningococcal varietal.
Yes.
And maybe that's not present in your oyster biofilms, but you know.
Yeah, and some of these don't give us the resolution to the species. So some of them, I mean, we just get genus and then we speculate and then we look at the matches and yeah, that's true. But I think I agree. For in terms of classes, I think I just put one pipeline up for that I thought was good. But I actually just ask my peers, my collaborators, what they're using and what's working for them. And I think if there's something that I feel like they feel it works for them, then I try that instead of exploring a whole bunch of pipelines out there. And so far that seems to have worked a little easier than.
So what I would do in the Neisseria thing was I would say, hey, great, we've got this data, maybe even got metagenomics data. And then I would say, well, where's a unique gene that this Neisseria meningococcus has? And I would PCR, save your DNA samples. If you really care, they're in the freezer, go back and do just a PCR. Do you get that gene? And then do then a real-time PCR to see if you have the numbers, because you can always go back to the DNA if you have it, and the gene it'll be in there somewhere. And if that's really it, it'll pop up. You could also do more assemblies. You could do some bow tie 2 analysis. There's some other things you can do that don't take that much time. But Archana, I was going to ask you about source tracker, because I don't know, you said you had a question about like where all the stuff was coming from the environments, like soils or sewage or all that. And I think I must have mentioned this to you before, but the source tracker, you have all the data you need to do a straightforward source tracker analysis with your students, because you don't even have to collect new data. You could take sewage, you could take fresh water, you could take 16S genomes or libraries from all of these things and ask, what's coming from where in those samples. So if you want to do that, please talk to me. And then in the future, you can collect samples from places you think it's coming from at the same time you collect your samples.
Yes.
And then I can say what proportion is coming from here, and maybe even which particular bugs are signatures of what's happening in those environments.
Oh, that'll be fascinating. And I see Gabby and Ben in this call, so we will be in touch.
Yeah. This is we had this for about 15 years now, this program, and it's built for 16S, but it can also do metagenomics. It can even do chemicals. So,
metabolites, yes,
yeah,
yes.
I can actually tell you metabolites. I can tell you chemical runout if you have that data.
Yes,
a little trickier, but it's very possible to do all that. So, and it can even do antibiotics. Well, where are the antibiotics? Yes. What's the source of the antibiotic resistance genes if you have the data?
Yes. Yeah. Oh, fascinating. This is wonderful.
We had it. Well, let's see. It's 1:03, and I just want to make sure. Trevor had a question in there I thought we could get to. Is there a de novo approach tool for AMR marker discovery? Or ARG, whatever you call it. And ARG is antimicrobial resistance.
Yeah, AMR.
And AMR is antimicrobial resistance.
There is a paper that I would put, I'll just paste that in the chat. It's by a team. I think they came up with this approach, but what they also do is they classify the AMR genes as high, low, or medium risk, I think. And I think this is good because we tend to just say all AMR genes are bad, but that does not have to be the case just because they are antimicrobial resistant. So this is a good paper because it brings perspective to what is truly a health risk versus what is not. And so I think that might be something that you'd like to look at, perhaps, Trevor.
I love that paper. Yeah, that's really great. I'm going to pop it up. Okay, well, it's already 1:04, and you know what I'm going to do, Arjun? If you have all the time in the world, I can tell you're not doing anything these days. So I'm going to send you a list of all the questions my students have, and if you
I would like that.
Like the ones you didn't answer, maybe they'd appreciate you writing these notes on the side. So, but if you don't have to do it, but they're good.
No, I'd like to do. Of course, I will. Yeah.
I can't get to them all.
Thank you.
You're such a great speaker, and you're so generous of your time. Sorry, I feel like I'm the introducing person. Everyone's supposed to do. Me.
Oh, I while she was talking, I actually pulled up this photo. This is us from high school. There's me back here, and then anybody want to take a stab at where our speaker is in this photo?
Oh Lord, this is a little fuzzy on my end.
I know.
Second from left, second row, but no, there you go. Oh, you're
right here. Yeah, isn't that crazy? Like, so this was, I would say this was what, 2002, Archana? Maybe 2001, 2002, right? Crazy times. I love it.
Come on, guys. It's very likely. India is a small country with very few people. It's
right, exactly.
Likely to be from the same high school, right? It's only 1 billion,
Isn't that crazy how time, just anyway, but worlds collide, and I love it. And I'm just so happy you agreed to give this lecture, because I think people really, anyway, we all learned a lot, and it's phenomenal, the cool stuff that you guys are doing. All right, well, I want to thank you so much, and
thank you.
We're looking forward. So hopefully other folks, especially on this call, hopefully you can join us every Friday, this fall at noon. Same Zoom link. And we'll see you next week. So next week, it's going to be Dr. Renaud Berlemont, and he is a bioinformatician from CSU Long Beach. And so he's going to be giving a talk. You'll see the flyer come out very soon about his talk. So thank you all. Yeah.
Thank you.
All right. Thank everyone for coming. Great turnout. Great turnout. Everyone's saying how great they love your talk here. So yeah, we're so happy to have you our first new season, and I have a lot to live up to now.
Oh no, I'm fascinated by your the mass balance thing.
Yeah, we should also talk about like CODA analysis, computational analysis. And also, are you counting stuff? I'm really excited about that. And also, like, how did you find the plasmids? Like, I'm doing antibiotics. I have 2 papers I need to send you. I'm doing sewage treatment. I'm doing the same kind of stuff, but with the Tijuana River. and it's
Yes,
nasty.
yes. I remember talking about the Tijuana River last time, and I don't think it's like every year we meet on this day.
I know, we gotta talk. And we also want to talk to you about a potential conference proceedings we want to put together with the CSU people. So.
Oh yes, yes. I will reach out after the in an email to you, and maybe we should connect over Zoom. Or, yeah.
Okay, we'll do that. I know you got to go, but
thanks, guys.
Thank you. This was lovely. Thank you for
putting this together this year.
Sure.
All right. Bye-bye.